For 60 years, a drug derived from the French lilac plant has been the global standard for treating high blood sugar. That drug is metformin. Millions of people take it daily, relying on a proven safety record that has made it the unrivaled first-line therapy for type 2 diabetes. The established medical consensus explains its success through the liver. For decades, textbooks have touted that metformin travels to the liver to suppress its glucose output, effectively cutting off the supply of excess sugar at the source. This liver-centric model defined diabetes care for generations. However, a closer look at the raw data reveals a biological assumption that is mathematically impossible. Scientists have identified a massive concentration gap between the laboratory and the clinic. There is a disconnect between how much drug a liver cell needs to respond in a lab and how much a human patient actually takes. This chart shows the concentration required to stop liver cells from producing glucose in a petri dish, massive millimolar levels, but the concentration found in a human patient sits in the micromolar range. That is thousands of times lower. Human clinical doses are physically insufficient to activate the anti-diabetic pathways scientists have studied in the liver for 60 years. Since the math fails in the liver, the drug must be acting on a much more sensitive control center located elsewhere in the body. Researchers at the Baylor College of Medicine recently published a study to track exactly where these low clinical doses of metformin were triggering a response. Their investigation moved past peripheral organs like the gut and liver, looking instead for a target that could coordinate systemic metabolism from a central hub. They tracked the drug's path and found that it aggressively crosses the blood-brain barrier, moving directly into the central nervous system. These findings confirm that the brain is the primary recipient of the drug at therapeutic doses. Specifically, metformin targets a region called the ventromedial hypothalamus, or VMH. This deep-seated control hub coordinates whole-body glucose levels through a complex network of hormonal and nerve signals. This neural circuit is roughly 1,000 times more sensitive to metformin than the liver, finally resolving the concentration gap. Within this region, the drug interacts with a specific molecular switch, a small protein called RAP1. Under diabetic conditions, RAP1 activity is pathologically high. It acts as a biological brake, suppressing the brain's natural ability to lower blood sugar. Metformin travels to the hypothalamus and inhibits this protein. It flips the RAP1 switch to the off position. Shutting down RAP1 frees a specific cell population called SF1 neurons. These specialized sensors dictate how the body handles sugar. Once active, they send electrical commands through the autonomic nervous system, signaling the liver and gut to lower systemic blood sugar. Rather than attempting to fix the liver directly, metformin activates a brain circuit that commands the body to correct the imbalance. Current metformin doses are high enough to flood the liver and gut just to ensure a tiny amount reaches the brain. This high peripheral exposure is why so many patients suffer from gastrointestinal distress. Identifying the RAP1 pathway allows scientists to develop a new class of central sensitizers, drugs engineered to target the brain directly. These future therapies could use microgram-level doses to achieve the same clinical results, bypassing the digestive system entirely. This fundamental shift moves modern diabetes treatment away from mass chemical flooding and towards highly precise, targeted neurological intervention. This discovery also explains why metformin appears to protect against brain aging and neurodegenerative diseases. Because the drug's primary mechanism is situated within the brain's own circuitry, its neuroprotective benefits are central features of its pharmacology, rather than accidental side effects. Researchers are now evaluating the RAP1-SF1 axis as a master biological dial for metabolic health and human longevity. By mapping these central signals, scientists have provided a specific biological roadmap for addressing the mechanics of human aging.